I'm very happy to tell you a little bit about what my lab's been working on for these last 4 years, with a little bit of background in there as well. My lab, as you can kind of see, we care mostly about neural tissue. And so when we think about neural tissue, we're mostly thinking about things within the central nervous system, right? And so the central nervous system includes the brain and spinal cord. And we care about this in the context of diseases and disorders of the central nervous system, or CNS as I'll refer to it, lead to a great deal of disability within the United States. So there's more than 10 million people in the country living with various different conditions and disabilities. Those span a wide variety of types of things. Those might be things that are genetic in nature, such as Huntington's disease. And so you can see here, this is just half of a brain. On the right, you'll see a healthy brain. And on the left, you see a Huntington's disease brain. And you can tell just from that, there's a gross difference in the size of the brain here. But you can see the gross difference in the morphology there of these sections of the brain. So that's an example of a genetic disorder. We can also think about this from the context of an injury, right? And in the case of an injury, we have aspects such as a spinal cord injury. And so you can see here, this is on now the tissue level. So you can see this at the organ level, but at the tissue level, we have this large cyst that forms. And so this is not useful tissue, right? So the spinal cord has essentially tried to wall this injured area off from the rest of the spinal cord. Right? So we can see at the tissue level. We can also see examples of this on the cell level. So here we see a small piece of tissue. And so these guys are individual cells taken from a normal brain and a Parkinson's disease brain, so a neurodegenerative type of phenotype. And you can see there's a difference here, right? So we see the loss of these guys. We might see a couple of them left. So these are dopaminergic neurons. So these are the neurons in the brain that make dopamine. And they're almost completely absent in this brain from a Parkinsonian patient. And the problem is, especially with things like Parkinson's, is by the time you can see physical symptoms of Parkinson's, about 80% of these cells are already gone. So the ability to reverse the disease is very, very much a problem. So I mentioned at the cell level, but at the tissue level and at the organ level, there's this common aspect, whether it be genetic, an injury, or a neurodegenerative type phenotype, where we see a loss of cells. And so these might be neurons or astrocytes or oligodendrocytes, but they're cells that die. And in order to improve upon the kind of physiology that these people are exhibiting, we have to figure out a way to replace these cells, because these are generally considered non-regenerative types of cells. So we think about this from the perspective of trying to engineer tissue that might mimic the native extracellular matrix, or ECM, within the central nervous system. So what does that look like, right? So you might imagine, sure, there are neurons there. They might also have some neural stem cells. There's a few of them, even in adults, contrary to what you may have been led to believe if you took biology 10 years ago. There's astrocytes, so these star-shaped cells. Those constitute the majority of the cell types within the brain. And you have oligodendrocytes, dendros, so a few branches from Greek. And these cells actually make myelin. And you may be familiar with myelin. It essentially serves as an insulating sheath on these long processes or axons from these neurons. So those are present. But all this exists within the context of an extracellular matrix, right? And that matrix might be amorphous in nature, or it might be fibrous, right? So you have these fibers that give guidance to cells to follow or grow along. So you have the matrix, but you also have aspects of various different factors, or growth factors, or proteins, or soluble molecules that can diffuse throughout this whole matrix, but also become attached to it in various different places. And so the matrix can mediate the presence or absence and local concentrations of those various different factors. So that's the kind of thing that we like to think about in my lab. And we like to think about it from the context of, if we take a neural stem cell and put it in some biomaterial of some kind, some matrix, what are we interested in doing, right? So I mentioned cells die in the context of disease, so we want to prevent cell death, right? That would be aspect number one, fewer dead cells, right? We also might be interested in stimulating the neural stem cells, the stem cell populations within the brain, to make more of themselves, because they have reparative capabilities. we might also be interested in using that material to direct the differentiation or the formation of new cells from those stem cells. So we might be interested in taking a neural stem cell, shown here in green, and driving it down to become a neuron. This is where a large area of neural development has kind of focused on. But we might also be interested in driving it down other pathways. So collectively, these cell types are called glia. And again, astrocytes, which are kind of star-shaped, and oligodendrocytes, which are those cells that make myelin. These are the cells that go haywire in the context of multiple sclerosis, for instance. Okay? So these are various different things that we might be interested in controlling. And how do we do that with a material? Well, we might mimic that native environment that I tried to give you an aspect of in the last slide. And so we need something that's hydrated and tissue-like. Your brain is very, very watery, right? Your brain is also the softest tissue, although I know there are other biomaterials within the body that might be softer, but it's the softest organ that you'll find within the human body. We might be interested in tuning the release of different bioactive moieties, growth factors, drugs, those soluble factors that I mentioned in the last slide. And being an engineer, we're really interested in having independent control of things like biophysical and biochemical properties. Right? We want to be able to finagle each of those independently as much as we like. That's an engineering mindset, right? So we'll take a look at this, looking at promoting neural cell survival and directing cell fate, looking at all these different pathways. And I'll tell you 3 different kind of snippets about work that we've been working on. One, using synthetic polymer gels. So we make these polymers synthetically that we use to direct fate through changing the mechanics and degradation of the material. I'll tell you a little bit about some microparticle drug delivery. We rescue cells from some reactive oxygen species or oxidation-derived injury. And then some protein-engineered polymer gels that we use to promote survival and then direct fate. So I'll tell you a little bit about each of these, starting first with these synthetic polymers. So another way to put this, right? So this is our lab mission statement. We think about this in the context of trying to use materials to control things about redox biology. Chemical engineers care a lot. We know a lot about redox. But we can think about this in the context of within a cell. A cell has a redox state, a balance between oxidized and reduced species within the cell. We think about drug delivery, and we think about tissue engineering. And I'll start kind of looking at the interface of these two. So our basic material, our most basic material that we work with in the lab is a polyethylene glycol base. So PEG here in green, it's a hydrophilic polymer. We can, if we so choose, add degradable lactic acid units. So we can put very short pieces of polylactic acid onto that, and then cap that with, in red, reactive methacrylate groups. So these reactive methacrylate groups allow us to do redox-mediated, free radical-mediated polymerization. So you can take this molecule, it's mostly PEG, so it's hydrophilic, it dissolves easily. We can dissolve it in aqueous media, cell-friendly media, add some photoinitiator and expose it to light, and we get a nice 3-dimensional hydrogel. So you can kind of see a representation of that here. So the cells are suspended within 3 dimensions within this whole hydrogel. And then in the context of some water and time, this will degrade into polymethacrylate chains, shown in red, polyethylene glycol, and lactic acid. So this little PLA bit kind of breaks up into lactic acid. So all those things are easily cleared by the human body. So this might be a good candidate biomaterial that we might be interested in putting within the brain or spinal cord. So a quick little bit of background about why we care about this material. When we encapsulate cells, so as a graduate student, when I encapsulated these neural stem cells within this hydrogel, and we looked at increase in DNA, so it tells us something about the proliferation of these cells, how much of these cells increasing over time. So this is after 1 week in culture, and we see we can change things about the macromer, the amount of polymer that's present, and this tells us a little bit of things about stiffness that I'll mention in a moment. But we only see a lot of proliferation in this one particular condition, which I believe here is 7.5 weight% of macromer. This is interesting. Cells survive over here, but they don't really proliferate much. And then once we get out here in very stiff, high macromer weight% hydrogels, cells actually die via apoptosis. That data I'm not showing you. But this is interesting. Why is it that one condition? All right? Well, if we look at the stiffness of the hydrogels we make from those different conditions, changing the weight%, you can see the compressive modulus, which tells you something about how flexible that material is. Right? the native tissue from which these cells were derived is about 3.4 kilopascals, right, in stiffness. This particular hydrogel that we made at 7.5 weight percent is about 3.7 kilopascals. So at least one factor that makes a big difference here is the cells that we grabbed from this embryonic brain tissue, when we put them in a material that matches the stiffness that they were taken from, they continue to develop and proliferate and make more neural stem cells, as we might expect them to do in vivo during embryogenesis, right? So this factor, that particular condition, did really well for proliferation. So stiffness matters in the context of neural stem cells, at least. Right? But there's another factor that might play a role here using the same biomaterial. We can look at the presence of free radicals within these cells. So this tells us something about the redox state. And we can actually stain for free radicals within the cells. And you'll see there's a lot of them here and not so many here, right? So these cells are in a more reduced or less oxidized. They got their antioxidant activity is doing a little bit better, right? And so you can tell that these cells are more oxidized. You can see really bright regions. These are the mitochondria within the cell, which is where most of your reduction oxidation happens. But when we encapsulate cells inside of a hydrogel that has that degradable component, right? So this is changing the degradable content of a hydrogel, all degradable, PEG PLA dimethacrylate, non-degradable, just PEG dimethacrylate, no polylactic acid degradable component. Cells have a lot different reactive oxygen species, so large differences in the number of free radicals that are present. In a degradable hydrogel, there's very few, right? And this is present immediately after encapsulation, right? Right away when you put the cells in there. It's continues at 2 hours, but it's also has somewhat of a chronic effect, right? As you continue to grow cells in these materials, we see happier, healthier, more reduced cells in these more degradable hydrogels, right? And we continue to look at this a little bit more, and there's an aspect of that polylactic acid. There's an aspect of pH that mediates that. But I'm not going to get into too much of that because that's quite old. But I wanted to give you kind of a little bit of an introduction of redox state, as we're going to continue talking about that a little bit more in the future. So free radicals in general, you probably have a pretty good idea. Free radicals are bad. They're evil, right? Antioxidants are good. Eat your blueberries, right? But and that's great. There's truth to that. But not all free radicals are bad, right? As is true for almost any molecule, the key is in the dose, right? So if you got rid of all free radicals entirely, your cells wouldn't be able to function. You'd have no metabolism, right? They're important signaling molecules as well. But too many of them can be toxic. All right, so we can measure one other thing. And so this is the last little bit of introduction I want to mention, this molecule called glutathione. Glutathione is a free radical scavenger. It is a natural, native intracellular antioxidant. So this molecule will scavenge free radicals. And the more of it you have in the reduced form, so it hasn't eaten up a bunch of radicals first, the happier and healthier we think of these cells being. And so we see more glutathione in the reduced state in these hydrogels that are more degradable, these 100% degradable hydrogels. And that's true, again, right away, as well as after a week in culture, right? So these particular types of hydrogels have cells that have fewer free radicals and higher levels of glutathione. Okay. All right. So when we think about this, we want to extend now, as I started my lab, beyond neural stem cells. So there's a lot of folks working with neural stem cells. We've learned a pretty good deal about how to control those kinds of things. And I thought, let's look a little bit more at these oligodendrocyte cells. So these are the cells, as I mentioned, that go haywire in the context of multiple sclerosis, right? And they're a very understudied cell type. So in order to get this cell, there's a progenitor cell in between called an oligodendrocyte progenitor cell, or OPC as I'll refer to them. And those cells are kind of this intermediate cell between a neural stem cell and an oligodendrocyte, right? So if we want to get an oligodendrocyte, we have to go through this guy first. So we can encapsulate these cells within hydrogels as well. And we see, not unlike what we saw with the neural stem cells, these cells expand. We see lots of good, healthy, happy ATP producing, metabolically active cells in these relatively soft hydrogels. So we're now looking at materials that are even softer than we were looking at before, more compliant. And these cells are distributed throughout 3D. They stain, so they're GFPs. They've been engineered to have this green fluorescent protein, so that's going to be present in all of them. We stain for nuclei with this DAPI stain, and then PDGFR alpha is an important marker of this oligodendrocyte progenitor cell. So they are what we say they are, is what we can say from this. And they grow in all conditions, but they grow the most when we have this most compliant or least stiff hydrogel. Okay? So just like with the neural stem cells, stiffness appears to matter. And when we encapsulate these cells are distributed in 3D. So you can see cells in this hydrogels are truly in a three-dimensional environment. They're absolutely distributed throughout the whole context of the whole hydrogel. Right? So that's great. We're giving them something that is like some kind of native amorphous hydrogel here. OPCs, as I said, respond to stiffness, and we looked at these cells are derived from a mouse line of cells, and we're looking at this condition of hydrogel. So these are all 7.5 weight % hydrogels, but we're changing the length of the macromer, the length of the crosslinker. So a longer crosslinker, you get more space in your net, right, and you get a softer hydrogel, right? So things can move around in there a little bit more. We see the ATP works there. We see the DNA cells seem to grow a little bit. And then we repeated the same set of experiments. So this is one oligodendrocyte mouse cell line. We repeated the same set of experiments to validate with a whole nother cell line. We saw the same basic effects, right? So we see more happier, healthier cells in these most compliant, these least stiff. So an 8,000 molecular weight crosslinker, 7.5 weight %. So this comes out about that 240 pascals. So very, very soft, barely a hydrogel, kind of snotty, very soft jello, that kind of thing, right? So cells like that, these OPCs like that. And we see they grow and they make these big clusters. This other cell line, they grow, they don't make as big a clusters. But this is also true in the context of cells growing in a two-dimensional petri dish, right? So we see similar things in 3D as we do in 2D, which is kind of cool. But we see these big clusters. So you can see them zoomed in a little bit more. So we have a live dead stain here. On day 4, lots of big clusters, right? And the least stiff hydrogel has the most big clusters, right? And we quantify that here, right? So the least stiff hydrogel, bigger average radius of our clusters. And then we also said, well, are they actually proliferating? Are they making more of themselves? And so you can do a stain for EDU. So cells that are actively proliferating will take up this stain, and they'll show up in kind of reddish here. And so we see lots of cells doing that in this softer hydrogel. So cells are proliferating. These OPCs are proliferating. They're making more. We can quantify the number of cells that are proliferating over here. There's a lot at 4 days, not as many at 7 days. That's also true in 2D. As you get more and more cells, they start to run into each other, and that's a signal for them to stop making more of themselves. So this is how, one way in which your body naturally mediates prevention of cancer, right? So they see other cells around them, and so they stop proliferating, right? So we see these kinds of things happening here. When we get a lot of cells, they start to slow down a little bit. So we have cells actively proliferating, right? And what about differentiation? Now, they're going to turn into the cells we actually want, which are those cells that make myelin, right? Those cells in the context of multiple sclerosis that go haywire. So we did a couple of stains. Again, we're looking for this particular stain. And you can see 3 different stiffness hydrogels. And we zoom in on each of these clusters. And you can see, it kind of shows up. But the red is what we care about here. This should be a membrane stain. It sticks around the outside of the cell, right? And we see that present in these 2 conditions, but not over here, right? So we don't see very much of this stain for PDGFR alpha in the context of this least stiff hydrogel. So what we actually think is happening here is that cells in this very soft, very compliant hydrogel have started to actually de-differentiate and go back up the progenitor cell stream, whereas these cells are continuing to stay as oligodendrocyte progenitor cells and making more of themselves, but staying on that kind of pathway that we're looking for between neural stem cells and oligodendrocytes. So the cells are continuing to do pretty well, but they're mostly staying pretty rounded up, right? You see them mostly as these kind of cell clusters with maybe a few branches starting to stick out a little bit. Excuse me. That's not ideal, right? That's not actually what we're looking for. We want those cells that have all those processes in that cartoon that I showed. Right? So how do we go about getting that? So we returned to this idea of a degradable polymer, right? So we have these polylactic acid units in here, right? So we have this degradable polymer, and we can add these two together to change the amount of degradability, right? So we have a 25% degradable and 100% degradable. And we can look at their metabolic activity, right? This is just another measure of metabolic activity. More equals better, right? And when we put them in the entirely degradable gel, we have more metabolic activity. Cells are healthier, happier, et cetera. Great. But what about that phenotype? What do they look like, right? And so when we have hydrogels that are both soft and degradable, now we start to see cells that are extending processes, and this is a hallmark of what cells should be doing in their differentiation pathway to make actual oligodendrocytes. So they have to do this process extension bit before they can start to make myelin. When they make myelin, it's actually these processes that reach out and wrap around, right? So we have to have this in order to get things that we want to see. If you make a stiffer hydrogel, higher weight percent, the cells are still stuck, right? But if you wait long enough, they will start to extend processes. But you see in this case, lots of tight, rounded up cell clusters. Right? If we now say, well, what if we give them something to pattern on, right? And so in here now you're seeing in red are fibers that we've just encapsulated into our hydrogel. So these are fibers they're just polystyrene fibers, so pretty easy process to make electrospun polystyrene fibers. A little bit harder to embed them in a 3D hydrogel along with the cells, but we did it, obviously. But you see the cells like the fibers, and those fibers were specifically designed to be on the size diameter scale of a mature axon, right? So these oligodendrocyte progenitor cells in green, what they normally do is they reach out their process and they wrap around an axon. But they should only do that when the axon is done growing, when it's mature, right? A mature axon is on the order of 1 to 2 microns in diameter. And so those are the kind of fibers that we made. And you can kind of see, we haven't done a lot of characterization. This is very preliminary still. But the cells are reaching out. They're certainly liking the fibers, and they're following the fibers in some cases. You can see a higher zoom magnification here, and a process that's reaching out and following these fibers and trying to reach out and grab other fibers, right? So we think that this context of topography, as I'm sure many of you know, is extremely important in directing stem cell fate. And this is just further evidence of that in the context of this specific cell type. So we're pretty excited about that and continuing to study that. But one other way, we said, well, that's not enough. What's another way we can try and guide fate? We can use some of those soluble molecules that we mentioned. So in this case, we can add this PDGF-AA. So this is a soluble growth factor. The GF stands for growth factor, right? And we can add that in, but if we control the rate and dose of that molecule via making these PLGA microparticles, so this is a pretty common material that you can use. PLGA is used for like degradable sutures, for instance. But we can turn it into microparticles and encapsulate this molecule inside of those and change the polymer to change the rate of release. So we have a slow release and a fast release, right? And one intermediate one. And when we do that, now we have our first evidence, right? If we control the kinetics in the right way, i.e., we actually withdraw this molecule very fast, right? So this is a classic example of something we didn't expect to happen, right? So our empty particles show us the most myelin basic protein. So that's the most differentiated cell was going to make this particular molecule, right? And so we saw the best development, the best differentiation when we did not have the molecule present, right? So the empty particles that weren't delivering anything, and when we added PDGF in the first media change and then never again, right? And so this was something that frustrated us at first because we said that can't possibly be true. But all these cells started with, before we did this experiment, they were all grown in this media containing this molecule, PDGF-AA, right? This guy down here. And so when we take them and transfer them to a new situation and remove the PDGF, that appears to be the cue to actually get them to start differentiating. And there's a lot of interesting, great work that's going on with this PDGF molecule in general that is well beyond my ability to do signal transduction and things like that. But this is maybe another piece of the story for these particular cells is this molecule, it's great for proliferation, but not for differentiation. We actually want to stimulate them to turn into happy, healthy oligodendrocytes that might make myelin. So the first bit here, just to summarize, right? So we have stem cells that can survive and grow in the context of these synthetic hydrogels. They make more of themselves, they proliferate, and we can, at some level, control that. In the absence of certain cues, they'll mostly grow as these kind of spheroids, these little balls of cells that proliferate but kind of stay in that local area. The ability to make more of themselves is dependent on the hydrogel stiffness, which you might expect, right? If you put them in a material that matches their native environment, they're going to do better than if you put them on a bone type of stiffness material. And ultimately, though, we think the degradation of that material, right? We need cells to degrade the material so that they can make their own extracellular matrix. That's the ideal. And topography, giving them something to pattern on, is going to be critical to the success of our future strategies with these cells. So to transition then to the second bit, we're now going to talk mostly about drug delivery, but still in the context of redox biology, right? So we're going to come back to these idea of glutathione and free radicals. So the molecule that I'm going to spend most of my time talking about here is N-acetylcysteine, right? So you can see an example of it here, right? It's a short peptide, and it has a couple of different things. It can be a direct radical scavenger, but it can also get incorporated into the synthesis of that molecule I mentioned earlier, glutathione, right? So this is a native antioxidant inside all of your cells. But this NAC, do any of you take NAC? Anybody know about NAC? Yeah, a couple people, right? So you can go pick it up at your GNC if you want. You know, it's like, I don't know, $60 for 100 grams or something like that. It's a cheap molecule, right? And that's actually critical to the story that I'm going to tell. But it will directly scavenge free radicals, right? So we said, well, we want to take this molecule. It's a small molecule, right? It's only a couple hundred grams per mole. We want to take that molecule and we want to put it in our PLGA particles. And usually the way you do that is a water, oil, and water emulsion, right? So you take your molecule, you put it in water, and then you mix it with your polymer and organic solvent, and then you take that and put it in another aqueous material, and you just sonicate the heck out of it, right? Small molecule. Small molecules, when they're hydrophilic like this one is, don't stay in that original water phase. They want to leak out into the second water phase, so they don't stay inside the polymer. But people have used this a lot, right? So you can change the polymer and you can change the release rate. And this is exactly what we want to do, but we want to do it with this small molecule, which is traditionally very, very difficult. Right? We can tune the lactic to glycolic ratio to change the release rate, and we did that. But how do we get this molecule in here? So we modified this traditional method and basically said every phase we're going to dump a ton of N-acetylcysteine into, right? So even that second water phase, which is usually like a wash step, like you're just washing away the excess stuff, we said we're going to put a bunch of N-acetylcysteine in there as well, right? So all we did is just dope every phase with N-acetylcysteine, and now rather than having 3 or 4% of our encapsulation efficiency, which is actually pretty standard for small molecules in aqueous phase that like water, we're seeing things that are 100% and better. Right? Depending on the polymer, right? So this is the ratio of lactic to glycolic acid. This is the inherent viscosity, which translates to the molecular weight, how long the polymer is. So we can tune those two things, right? But mainly we said, all right, we have real measurable NAC that we've managed to lock into these cells, right? And we can tune the release rate just like you would normally expect, right? So we can change the properties of the polymer and we can get the N-acetylcysteine to release at different rates. We see release at least out to about 7 days or so, and this one continues to release for a period afterward. Right? Then what do we do with those? Right? The whole point is we wanted to scavenge free radicals. N-acetylcysteine is an antioxidant. That's what we want to use it for. So does it scavenge free radicals? So we exposed it to a bunch of free radicals, and we said, is it scavenging? So we took the supernatants, the stuff that's released from those particles, and yeah, we can scavenge radicals. So supernatants, even released at 7 days, are still scavenging free radicals. So there's great antioxidant activity there. Right? And does this protect cells from any kind of damage? That's the thing. That's the key point. Right? So we set up a whole bunch of assays. So we use these oligodendrocyte progenitor cells again. We expose them to free radicals. We use hydrogen peroxide in this case. Right? Naturally decomposes into peroxide radicals and hydroxide radicals. And we said, if we add these particles that release NAC, can we protect cells? Can we rescue cells? So we had a whole bunch of different treatments. I'm only going to talk about one. The rest of these are all published already. But this particular one, where we take cells and we give them these particles, and then we hit them with peroxide, multiple doses over multiple days, right? We thought this would be most similar to the context of an injury environment where you're constantly having production of oxidants, and macrophages are constantly releasing things to kill cells in the context of inflammation. So we said we thought that would be most similar. So that's the one I'm going to concentrate on here. So we have cells, right? We're looking at the relative viability of these cells. If you just give them peroxide, the cells don't like it, right? So right after you give them peroxide, they're mostly unhappy. There's very little metabolic activity. That's what we would expect. If you let them just sit for a day, right, they recover a little bit. They get some of their metabolic activity back. But if you protect them by adding N-acetylcysteine just in solution or as delivered from our particles, right, we're able to dramatically improve the viability of these cells almost back, right, so somewhere around 75% or so, almost back to a native, happy, untreated cell that hasn't been exposed to free radicals at all. Right? And you can see pictures of this, right? Healthy control, injury, cells are all like manipulated and dead and gone. You add this NAC, we see happier, healthier, and more cells present. Right? And we can look at this, just look at their morphology. Again, these oligodendrocyte progenitor cells, these are in 2D, should have these nice processes extending out, right, which are shown up here in phalloidin in red. Again, the cells are positive for green fluorescent protein. So happy, healthy cells unexposed to peroxide. Exposed to peroxide, they start to ball up and pull in those processes. If you add NAC in solution, so free NAC, they do okay. If you just add particles that we didn't give any drug, they don't look very happy either. Right? But if you deliver that drug from a particle, the cells do okay. Right? And we quantified all sorts of stuff. So we looked at, again, ATP divided by DNA. Cells look great, right? When you have particles back up to a healthy control, where we didn't expose them to anything at all, right? We also looked at things like nuclear particles per cell. So as cells start to undergo apoptosis, they fragment their nucleus, and they make little bits of nucleus inside of a cell. So the more fragments you have, the unhappier, the more apoptotic effects, more cell death that you're having, right? And so that's what we see in our injury conditions, but we can rescue that when we deliver NAC, our antioxidant, from a particle, right? And also we can look at nuclear cross-sectional area. The larger the nucleus, the more unhappy the cell, right? And so you can see, again, injury cells have large nuclei. When you add peroxide and just particles, no drug, large nuclei. But when you add drug, we're able to rescue those cells and keep them in a more happy, healthy state. So just to summarize this little bit on antioxidant delivery, right? So we have encapsulation efficiency. We have a new method that's now been published that can tell you, you can read all about it, about how we can dramatically encapsulate greater amounts of NAC. We have a preliminary patent for it. It's relatively something that is not hard to do, but it can only be done, caveat, with cheap molecules, right? Because you have to use so much of it, right? NAC happens to fit the bill really nicely. Dope particles absolutely scavenge radicals and protect cells, right? Bullet point 2 and 3. And we can do that better than just free NAC in solution because you have a constant release of that molecule over time as opposed to a whole bunch of it all at once, right? That's why you have to take medicine multiple times a day. If you just take it once a day or once a month, you get a big spike, and that has negative consequences, and you don't have that sustained over time. All right. So the third story I want to tell you about is focusing very much on this area of tissue engineering, right? And we think about this encapsulating cells in three-dimensional hydrogels in the context of an engineered protein, right? So far I've talked all about synthetic polymers. My lab also does a lot with engineered proteins, right? And the reason we care about this is because a protein can both be cell instructive and cell responsive, right? And we think about these proteins not too much unlike we think about polymers. They are essentially block copolymers. And the blocks that we care about here are maybe we want a bioactive block and we want a structural block, right? And you can see examples of what those might be. And so we engineered proteins that have the blocks that we want. And we use a structural block that is based on elastin, right? So elastin is a protein that's found in the human body throughout a variety of different tissues, but you can think about it maybe where things are more elastic. So you find a lot of elastin in your blood vessels, right? That have to pulsatile allow expansion and compression, right? So an elastin-like block, and you can see the sequence here. It has this repeat structure, VPG, and I'll say XG, right? And every once in a while, we'll have an isoleucine there. Sometimes we'll have a lysine, but mostly isoleucine, right? So this protein is cool for a variety of reasons. So we have this elastin block, and then we also have this bioactive block. So why do we want the bioactive block? So that we can tune the number of interactions that this material will have with the cell, right? So in this case, our bioactive block is a molecule that has this extended RGD. This RGD, arginine glycine asparagine, is cell adhesive, right? So cells will bind to it via integrin-mediated binding. And we can make the exact same protein and just flip these two amino acids to make a version that has RDG, right? And cells will not bind to this, right? So we can make these two proteins, we can mix them up together, and we can control the presence of RGD in this hydrogel. Great, right? Why the elastin bit? It doesn't have any bits that cells adhere to, right? So we can change this and keep this the same, and we can control how interactive the cells will be with it. ELP, elastin-like protein, or ELP, also produces pretty well. So if you think about fermentation, you can make a lot of ELP relative to other recombinant proteins. So that's nice. So you can make a whole lot more ELP than you can recombinant insulin, for instance, partly because it is a repetitive structure. It's been engineered very, very nicely. But the purification bit is also really cool. So I was talking with someone this morning about LCST, or lower critical solution temperature. So this behaves the opposite of most proteins. As you warm it up, ELP doesn't like it. ELP will crash out of solution and make a polymer-rich, protein-rich coacervate at warm temperatures, right? So it'll precipitate out, and most other proteins will get more soluble as you warm them up, right? So we can basically use that to our advantage to do production rather than the typical chemical engineering unit op of chromatography, right? We can just heat it up and centrifuge it, and then cool it down and centrifuge it, and alternately keep the precipitate and the supernatant, right? So that's great. Simple purification. That is nice. And then we've engineered it so we have site-specific cross-linking, right? So as I said, we manipulated it a few places to have this lysine here. So lysine gives us a primary amine that's a handle for us to do various different chemistries. So we can use that to cross-link our material using a amine reactive cross-linker. So we can do that. We can mix our protein with our amine reactive cross-linker, which is tetrafunctional, and we can change independently, like good engineers want to do. We can independently change mechanical properties, such as mass swelling ratio, for instance, while manipulating the concentration of that cell adhesive ligand, RGD. No change in the mechanical properties, big difference in RGD. Alternately, we can change the mechanical properties by changing the ratio of cross-linker, but not change anything about the number of RGD ligands that are present and available to the cells. So we can independently tune those two things. Great. We can encapsulate cells inside of it. So this is a dorsal root ganglion. So this is a little ball of cells that's found at the, it's technically peripheral nerve, but it's right at the interface with the central nervous system along the spinal cord. So it's a little enlarged piece of tissue. So this was the original piece of tissue. We encapsulated in one of these ELP hydrogels, and it extends processes. And this is a depth projection. So things that are close to you are kind of blue. Things that are farther away are red, which never show up very well on projectors. But you can see that there are processes extending out into this three-dimensional hydrogel at many different focal planes, right? And those processes that are extending are primarily neurons. So we have lots of good outgrowth. we get processes that are extending the way we would want them to. Right? So we have lots of outgrowth. And we can quantify that outgrowth over time. This is old work, so I'm not going to spend too much time on it. But the stiffness of that elastin-like protein hydrogel now, so not a synthetic hydrogel, but an engineered protein hydrogel, entirely protein with that little cross-linker, and cells, these dorsal root ganglion, they extend processes at a rate dependent upon the stiffness of the hydrogel. Stiff gel, cells don't extend. They're actually not very happy. Compliant hydrogel, we get lots of process extension. Right? And we can quantify this. We developed our own image processing algorithm to measure lots of neurite processes, so a lot of neuron J and image J type things. You can measure one process really accurately, but it's hard to measure hundreds of processes quickly, especially when they're really dense like those images. And so we quantified that, and we see statistically different population distributions in lengths of these processes. So this is a histogram, right? Even at 1 day, 24 hours after encapsulation, we get more longer processes when we have a soft gel. Right? And this is also true at 3 days and 7 days, where now we see processes that end up being millimeters in length. Right? So that's a hallmark of neurons. They can extend these axons millimeters in the human body. And so we're seeing things that are starting to get close to that in these hydrogels. Similarly, we can change the RGD ligand density. So when you have lots of RGD present, you get lots of outgrowth. If you don't have any RGD, you don't get much outgrowth. Right? Well, like I said, I wanted to cover that pretty quickly, because that's old work. In my new lab, we thought, well, how do we make this in a way that might be even more specific to the brain and spinal cord? Right? How do we make materials that we care a little bit more about? And we said, this protein laminin, there's a lot of laminin in the brain and spinal cord. That's something that is relevant specifically to that type of extracellular matrix. Right? We don't see a whole lot of RGD. Right? So laminin is interesting. You can see the structure of laminin here. It's a trimer. Right? It's huge. Right? It's a huge amino acid. It's a huge protein. 900 kilodaltons. Right? But it has lots of good functionality. Right? And so we actually focused in on this laminin globular domain, LG, and it's 1, 2, 3. It's the fifth one, so LG5. We focused in on that because that little module alone is about 200 amino acids or so. It's a little bit more approachable, but it has lots of functionality. It binds heparin, it binds alpha dystroglycan, and it can bind via cells via an integrin beta 1 binding site. So we said, that's what we're going to go after. Let's try that. And we said, let's make it, and we can use that elastin functionality to give us our building block, our structural building block, and use the laminin. And so we said, what are our criteria? We have a whole bunch of criteria. We want it to be thermodynamically stable. We want it to be bioactive. We want it to self-assemble, cell-compatible gelation, of course. We want to allow cell modeling, right? And we said, well, that's a long list of stuff to do, and I don't know if we're going to solve this at all or if we're going to have any success. So let's try and simulate it. Let's try a computational approach to see if this material will have at least this first property that we're after. We're pretty sure it'll be bioactive and such. Right? But let's see if we can go after that. So we designed this protein, and you can see bits of the sequence here. So we have this laminin globular domain, which is the majority of the protein, and then this ELP, we'll call it a tag, that we've stuck on the end of it. Right? So the laminin globular domain, again, has these functionalities embedded in it. And so we did a computational simulation, all-atom molecular dynamic simulation. And so here you can see the laminin globular domain. It's very stable. So each one of these wires is a time point in the simulation, and there's not a lot of variability here. So you see in yellow are the beta sheets, and you see highlighted in orange or pink, right? This is the alpha dystroglycan. Then here's the integrin, and then here's the heparin in purple back behind here. And this ELP region, which is just a single simulation here, right? So you don't see the flexibility yet, right? But this shows you the flexibility of that LG domain, right? So we said, take this protein and stick it in a water box, right? Like a good protein simulations person is going to do, and let it run, right? And let it run at a particular temperature, okay? So this simulation is at 310 Kelvin, right? So close to physiological. And this laminin globular domain, you can see it's not really moving much, but this elastin-like protein domain is running around like crazy, super flexible, right? And you can see in some places, right, some extended beta sheet showing up, right? So rather than just being random coil or alpha helix or beta turn, we see this guy show up, and it's pretty persistent, right? And that, we think, is super important for the self-assembly aspect of this material, right? If you can get organized structure, which a beta sheet structure inherently is, we think we can get things to kind of stick to each other and kind of self-assemble. And again, you can see the sequence up here. So we've had that VPG XG repeat sequence, and we have some lysines, some leucines, some isoleucines kind of extended throughout there. Right? And we chose that sequence because you can use those X's, those isoleucines and leucines, to vary what the lower critical solution temperature is when we think that beta sheet will show up. So we do the simulations. This is what it looks like at the start of the simulation, right? And you run it at 290 K, so well below physiological, and you kind of see what it looks like there. You run it at 295, so these are final snapshots, 300 Kelvin, right? And then we get close to physiological temperature, and now that's the only place where we see this beta sheet, this well-structured, well-ordered secondary structure show up in this protein. And you can kind of see very specifically, we actually have two of them here and one over here. Right? So this secondary structure we thought was important. So we quantified the secondary structure, right? So you can quantify it in these 5 bins, right? And you can look at this, the number of times that structure shows up at any given temperature, right? So this is at 290, well below physiological. And you see not much of this higher order, nice organized structure showing up here, right? But we do see it showing up when we get to these middle temperatures, right? These physiological temperatures. Lots of beta bridge, lots of beta strand. And these are the kind of things we're looking for. We think those are indicative of a molecule, right? These are all single molecule simulations, right? One protein, one several hundred amino acid protein. But we think those are really important to get these molecules once we put multiple of them together to assemble and have good structure, right? And then you can see this is the temporal evolution of that ELP area. So things are early are red, middle is gray, and then late is blue. So you can see how this changes from a really extended floppy thing, and it kind of coils up a little bit, but stays pretty open. But at these other temperatures, it coils in really fast, right? So this is a high temperature. At this property, that protein's probably becoming thermodynamically trapped because it's heated up so high, it gets stuck in this very tight structure. Whereas it takes a little longer here, but ultimately gets to something similar. And we looked at that general size by a thing called radius of gyration. And you can measure basically the radius of how big that thing is. And that collapses in all cases pretty quickly, but it continues having some flexibility in some of these conditions. We took one of these conditions at the end, right? So here in 310 Kelvin, it kind of peaks back up again. So we took that one, that final structure, and ran it again for another 100 nanosecond simulation. And that one continues to alternate, right? So it's not stuck, and it's not wrapped around the laminin, which is really helpful. But if you take that structure, this guy right here, and run it, change the temperature, it collapses back down again, right? So we're pretty confident that that temperature, for some physiological reason, is super important for the secondary structure of this protein. And then one last little bit, this is a little bit difficult to explain, but it's a contact map. So it's basically measuring the contacts between molecules on the protein. And so you see the residue number. So we have 0 to about 240 amino acids on this axis as well on this axis, right? So in blue is the laminin domain interacting with itself, right? And in red here is the ELP interacting with itself. And so what we're really looking at here is this area and this area. So we see these well-defined like box structures, right? So these are the beta sheet structures that are forming, we know exist in that laminin globular domain. We saw all those yellow, nicely aligned things, right? And if we look at different temperatures, again, 290, 300, 310, we don't see a whole bunch of contacts that are permanent here. So we don't see that great structure. So we know that these regions of this protein are not just gluing themselves to each other, preventing intermolecular interaction between different molecules. So that's a good box to check as well. So we have some potential with this. So quick summary of the proteins then, right? So we have a sharp change in that secondary, that important beta sheet structure happens at a particular temperature or close to that. We've observed then, we correlated that to an apparent LCST behavior, right? So we see a higher radius of gyration, higher order structure. We don't see, in this region, this LG and ELP regions kind of just gluing themselves to each other. But I think the broader area here is that we can use this computational approach, we think, to apply it to the design of new engineered extracellular matrix molecules. It's a challenge because these molecules are really large, and so they're computationally intensive. But I think it is an approach that is possible, especially if you have a good high-performance computing cluster available to you. So then the take-home message, right? So I talked a little bit about redox biology, drug delivery, and tissue engineering. We hope to use these kind of strategies to repair the brain. OPCs are an important and understudied cell in human disease, so I'm one of not that many people that are really caring about this particular cell. But it's something that has to regenerate in the context of any kind of neural injury, whether that be stroke or multiple sclerosis or Huntington's disease. We have to be able to replace this cell as well as the neurons. Engineering design via both theory and experimental synthesis can really provide different types of tools, polymers, proteins that are good matrices suitable to make tunable and engineered materials, and that we can use matrix properties dramatically to impact the various different cellular phenotypes of cells. So I'd finally just like to thank my students, particularly Lauren Russell, who's over here, and she led all of our polymer work with the oligodendrocyte progenitor cells. Nick Murphy, who is, where's Nick? He's the clown over here. He did our microparticle delivery work with N-acetylcysteine. And then James Tang, where is James? James is here. So he did all of our protein simulation work and is also now working with some peptides and making materials. I also want to thank all of our collaborators who were just starting to get some of that work out, as well as our funding sources. So with that, I'd be happy to take any questions.